First of all, I apologise for Brexit. It had nothing to do with me. I went to the US, all right? I went to the US and I thought people were looking after our country while I was away. So anyway, so don't blame me, okay? This is my technique for getting the crowd on my side before I start to talk. Okay, so another apology. I've got a lot of slides, but I'm going to put you through it. Okay, that's the idea. I want you to get your money's worth out of this course, right? But also it might fire off a few ideas about the cytometry involved in microenvironment analyses that you can sort of get back to me in some way. It's very easy to mail me. You know, you'll find me on the web or whatever. So any questions? Any support information or giving you any information in terms of slides, just email me, okay? Right, so that means you can go to sleep effectively and just ask for the talk, I guess. Right, first of all, I'm going to try and work through microenvironment cytometry in a slightly different way. I'm going to take it from the point of view of trying to develop drugs and targeted therapies that exploit that environment or in some way understand the limitations of that environment. That's fine. Okay. And so what I do, I thought I'd start off a little bit differently today, right? So and you'll see this in the next two slides. Okay. So this is a stitched together image, actually quite high resolution, although it's not obvious from this because you can zoom into that image, using a gigapan imaging system of a prostate cancer, supposedly, needle core biopsy. And what you see is the variation of tissues around that stromal elements. I thought it would be useful for us to try to look at those tumor microenvironments, those tumor microcommunities, and what some of the biophysical and cell surface properties and behavioral properties that make life in those microcommunities a real challenge for therapy. So, the theme of the talk will be experimental anti-cancer therapeutics, but I'll be taking little diversions, a little like the way that Andrea drives around Modena, little diversions here and there to touch upon the cytometry and its use and its applications. So, the two sub-themes are cell-to-cell and ECM interactions, obviously something important in those microcommunities, and then how can one perhaps exploit the hypoxic environment that's resident within those areas to target the cell cycle, introduce new classes of drugs into those environments in a safe way. Okay, so it's drug design and cytometry. So let's look a little bit more closely to that microenvironment as we zoom in onto that prostate cancer. Believe it or not, that's what it looks like as you try to zoom in. Please note that this is Machu Picchu, all right, okay. It's not a prostate cancer, but just in case you were wondering. So, as we go a little bit closer, you'll actually see that there's a micro community embedded within that image, and it's distant from its vascular supply. You could probably notice that there are larger organ structures within which it operates, almost certainly exocrine and endocrine structures. But here you can see there's also a hypoxic environment associated with that. Let's zoom in a little bit more closely. You start to see there's real structure, physical structures, biophysical structures within those microcommunities. A little bit more closely, you'll actually see the live stroma. And in some cases, you'll actually be able to just about see on this image here a cellular community. Apparently, similar, but relatively rare at this point. So as we zoom in a little bit more closely, you'll see that there are collagen fibers associated with this structure, and what appear to be migratory roots as well, probably leading to endothelial structures. We go in a little bit more closely. This is where it starts to get personal with the cancer cells, by the way. You'll see that there are biophysical and structural limitations to the ability of those cells to migrate or invade local areas. And this is actually quite a nice sort of metaphor here for the... Yeah, this is fine. This one I think is dangerous. Okay, obviously bought in China on the internet. I think this is safer, thank you. And these are sort of energy gradients, really, in which the cell has to move against pH, hypoxia, and also biophysical interactions with the structures to enable it to perhaps migrate and invade local communities. There are migratory routes. If anybody's seen macrophages chaperoning tumor cells along collagen fibers, it's absolutely stunning to see that in live videos, the fact that these are dynamic processes. As we go a little bit too personal, shall we say, too close on this structure here, we'll see that there are actually individual cells, and they're different in different ways. And there are subpopulations here, a little bit fuzzy as you can see, that may be sort of migrating along those routes and ready to actually perhaps contribute to circulating tumor cells. There's a resident community, that almost certainly the primary tumor that you're effectively trying to target with therapy. You go a little bit more closely still, you see that some of these cells are actively monitoring their environment, and actually, and this one here, for example, is actually trying to communicate, probably changing email addresses or something, I don't know, actually trying to communicate and interact. But look, there's at least, what, 20 nanometers, 35 nanometers between these cells here, because that's how cells interact. They stand off each other, they don't fully form a contact, they stand off by about 20, 35 nanometers to allow homophilic and heterophilic interactions with cell surface receptors. So it's little like a colony of porcupines, really, if you think of it that way. They not only interact, for example, you find out that there are targeting opportunities to disrupt these interactions and therefore the health of those tumor cells. If we go really close in, you'll see this XY cell. Obviously looks pretty fit to me, although, you know, I don't take notice of that too much. The selective environment has obviously enabled this cell here to, how should we say, be very fit for its environment. Almost certainly apoptosis resistant. It displays resistance to adverse environmental conditions, not least radiation. There's the radiation protection there, almost certainly because of changes in DNA damage sensing and also repair capacity. You'll also see that it's a CD marker, what we call a collage decal. A CD marker on the surface here, which displays, you know, distinct markers for us, for the cytometry community, to identify, if not target, and we'll get to that. They also have an enormous capacity to respond to stress within the system. And sometimes, for example, if this is an enhancement of P450 profiles within the cell, it will actually, you can actually use that in some way to target agents to that, and we'll come to that. Not least, if you look very closely here, it regulates its own cell cycle time, almost certainly without telomere restriction. And if you look very close, I think you're quite impressed by how we've got right down to this single molecular event here. I hope you're impressed, because it took me a lot of time to do this. The potential for targeting the cell cycle is there. One of the fundamental limitations on whether or not a cell proliferates or dies. And so we're going to be looking at this sort of these cells in their microenvironments, understanding how they interact at the level of the glycocalyx, the pH environment issues, cell attractors, that concept that cells live within a basin of thermodynamic stability that can be changed to another basin of thermodynamic stability. And once they've shifted, right, they're stable in that state. So attractor states in tumor cell systems are those in which they tend to show particular, for example, CD markers or a particular type of metabolism. But when challenged, there's a finite possibility to move into another state. And it's this, shall we say, phenotypic plasticity that's a real challenge for therapy, because the therapy itself can shift cells into different states that are then resistant to that therapy, as you'd expect, not just by selection, but by actually switching those states. Okay. So, let's get back to real cells now for a moment. So, obviously the stroma is an important component, an increasingly important component in understanding the microenvironment, the tumor microenvironment, almost certainly because of a whole series of reasons, such as there are reciprocal and heterotypic, different types of interactions between tumor cells and the surrounding stromal elements. In fact, there's a concept, of course, that it's better to target the tumor stroma rather than the tumor itself, because you know a little bit more about the biology, right? And that supporting stroma itself, once disassembled, the tumor cells lose their nurturing niche, shall we say. It's a competitive environment, and that environment is never, full stop, never replicated in any in vitro or partially in vivo system, such as a PDX model. That's because you cannot reproduce the dynamic nature of that interaction. All you can do is look at components. And that is why I think that many models actually fail to deliver drugs in screens. The tumor cells, the tumor populations may be very tissue specific. A good example of that is the stellate cells in pancreatic cancers that start to reorganize the collagen fibers, producing one of the most drug resistant and in fact deadly tumors available for analysis. Therefore, the ECM, the actual structure of this environment, can actually be modified by the tumor cells themselves by changing, for example, collagen signatures. And there are lots of techniques to look at that. One of the most elegant ones is second harmonic generation, of course, where you can just by direct microscopy see those structures changing. The bottom line is that almost certainly there's a whole range of therapeutic agents currently used in which the stroma itself is inducing the drug-resistant property. So let's think about the types of expressions that are occurring within, for example, just as an example, myeloma cells interacting with stromal cells. Look at that. The opportunities for targeting there are enormous. However, the opportunities for modification, secondary pathways taking over, are also enormous. Therefore, that specificity for therapy is virtually never achieved. And in the talk, I'll be touching upon the cytometric challenge of, for example, looking at cell cycle targeting, in particular using a class of drugs that target topoisomerase II, and also what this means, for example, for hypoxia, where I'm sure you're all aware there are a whole series of hypoxia response genes that are switched on under stress, not least HIF-1 alpha. So, for us, in cytometry, one of the challenges is, of course, how to measure these things dynamically, virtually never done. You can obviously monitor soluble molecules that are coming off. We've heard about this during the week. Membrane-bound mediators, and also mediators that can be shared by both tumor and stroma. So I'm going to focus now on the glycome. The glycome is the array of glycosylated molecules on the surface of a cell. The complexity is absolutely enormous. It's virtually beyond most analyses these days. However, there are certain molecules which, under glycosylation, start to significantly change the ability of a cell, for example, to migrate. And these effects, therefore, are enormous, whether or not they adhere to a substrate. In vitro, of course, you start to select for this just by growing cells on plastic. You start to completely change that glycomic array on cells. Very important to know. Cell-cell contacts are changed, matrix interactions. So if you're just working in Matrigel, you're in a completely abnormal but at least accessible environment, which has again changed that glycomic array. Whole series of issues there. So we'll be looking at, specifically, as a good example for targeting, the polysylation of neural cell adhesion molecule. So I'm just going to tell you a little bit about that. highly relevant to a whole series of neural crest origin tumours, not least neuroblastoma and small cell lung cancer. So, NCAM is a part of the immunoglobulin superfamily. Here we have it. You'll see the other members that you're familiar with. Importantly, the sites for glycosylation, and this is an extensive glycosylation, are specifically here on the molecule in its various isoforms, straddling to different extents the cellular membrane. And it's the glycosylation here, the addition of negative charges to this molecule, that can change its function. Interestingly, of course, if you look in the adult, there's virtually no polysylation. If you look in the embryo, you see extensive levels of polysylation on target cells. And that's because cells have to have plasticity within their environment for organogenesis, migration, for forming neural networks, etc. In fact, really the only time you see extensive changes in polysylation in the adult is during neural plasticity, when there's an intermediate level. This immediately tells a drug targeting person that if there's very little polysylation on NCAM in the adult, it means it's a restricted target for therapy. So, what does polysylation of NCAM actually do? Well, you can by physical studies, you can measure the interactions between cells that are polysylated or not, or different levels of polysylation. And here in this diagrammatic representation, we have force versus the distance between cells. And importantly, what happens as PSA is added to NCAM, it repels the membrane surfaces, whereas if it's removed, they start to oppose each other a little bit more closely. But look at the tolerances here. Normally cells are about 40 nanometers apart if they're not interacting. They're about 30 nanometers for interactions between surface receptors, and they're about 20 to 35 if you really have true engagement of most of the receptors. These are very small changes. You will not be surprised that in vitro culture in different systems starts to change those interactions, and therefore your ability to see what is really important. So, importantly, even though PSA is like the permission molecule for allowing cells to interact with each other, it's not necessarily the NCAM that is actually active in this. It's really sort of like, as I said, the spines of a porcupine, when really all the interactions are within other receptors, shall we say, that are being opposed in this system. That's important to understand. So, allowing cells to stand off from each other, or even the extracellular matrix in different ways, driven by NCAM in this situation, allows the permission for other interactions between adhesion and migratory molecules. So that's what a sort of diagrammatic representation of NCAM looks like, with its intracellular domain, the cell membrane, and antibodies here used to identify, for example, whether or not this molecule is polysylated or it's actually NCAM that you're looking at. So you can use these in combination to look for the state of polysylation in these molecules. Any carbohydrate chemists here? Hands up? No? Good, because I'm not, right? I've got a very strange friend who is a very good carbohydrate chemist, and you know, you don't want to meet these people. It's just, they're strange. However, it's important to understand that the glycosylation of many molecules is driven by the nucleotide sugar generation pathway, not least the conversion of cytidine through monophosphate, diphosphate, etc., going down through this pathway here. You see also that UDP can actually go into this system as well. So nucleotide sugar generation, synthesis, is the key to all of this. So look at the complexity there. There are hundreds and hundreds of enzymes just trying to produce different mixtures on the glycocalyx of different types of carbohydrates. It's obviously evolutionary, highly redundant, but there's a massive buffering in that system as well. So you try to change something here, something else takes over. Not least, there are negative feedback loops as well, as you can see here. However, when you get down to the point of polysylation rather than just silation by itself, you get this. It's driven by two enzymes. So we've got a target which is only really expressed in adults when there's a tumor. And you've got a target which is only driven by in its biosynthesis pathway by two enzymes, STX and PST, the polysilyl transferases. Perfect situation for molecular targeting. So let's think about what this means in terms of polysylation of NCAM for a tumor. If we take small cell lung cancer, which you'll realize comes from a neural crest origin, one of the features clinically for small cell lung cancer, of course, is that they generate neuropeptides. A tumor in the lung that's producing neuropeptides and producing, shall we say, syndromes associated with those neuropeptides. That's because of the neural crest origin. And as a result, there's early metastasis of this drug because of their phenotypic plasticity. They rapidly acquire drug resistance because of their ability to cope with the microenvironment. And they express PSA-NCAM at a level which is commensurate or equal to the extent of metastasis. So highly metastatic forms are highly polysylated. A whole series of therapeutic approaches are taken at the moment, but none of them to, shall we say, targeting on one of the main principles by which these cells can invade and migrate. So small cells they're really difficult to work with. I don't know if has anybody grown small cell lung cancer here in the, don't do it. You'll waste your life. They show substrate independent proliferation, and the adhesion process is thought to be controlled by PSA-NCAM, but we can show it's not, right? But migration is very important. Therefore, let me give you an idea what these beautiful cells look like. And this is one of the reasons that they're not included in the NCI drug screen, because they're so difficult to work with. So here you have a large cell which is behaving itself a different type of tumor cell of the lung. Flat, just grows as a monolayer. Everybody's happy in terms of in vitro culture. But small cells, either in extensive culture or directly from a biopsy, form these multicellular masses, their own microcommunities in 3 dimensions. Now this is important because it means that you can't do assays such as cloning assays very easily. You can't even do typical invasion assays because you put the cells down on a matrix to look for invasion and they all get together as groups. There's a natural herding instinct, little like British football fans abroad. You know, they're nasty, and they herd together. Right. So this is one of the problems, but these are ideal opportunities to look for microenvironment, micro-community interactions. I hope you realise that because they're already forming these communities. And what's driving that? An important thing about small cell, which is about 10 to 15% of lung tumours, but very therapy resistant, smoking induced, as you can expect, is that they show phenotypic plasticity. And in fact, when a histopathologist or pathologist is trying to diagnose this tumour, sometimes they think it's a lymphoma. Oh, maybe it's not a lymphoma. Maybe it's a squamous cell. Oh, no, it's not. It's a small cell. Right? Real difficulty. And that's not because there's infiltration of different tumour types. It's because there's massive phenotypic plasticity. This tumour can appear in different ways at different times with what appear to be different markers, right? This is, therefore, it's a moving target in terms of therapy. One of the questions, of course, is polysylation, given its embryonic role to allow plasticity involved in this? And can you stop that by inhibiting the polysilyl transferases? Does that make sense? Yeah, okay. I wasn't really asking for an answer there, by the way. Otherwise, it's wasted time. Right. However, if you just do a simple bulk analysis of polysylation looking at adherent versus non-adherent cells, right, you see that, look, adherent cells have lost their polysylation of NCAM. Very nice. Then you go to another line and it's completely opposite. Then you go to another line and there's no correlation at all between adherence and non-adherence and polysylation. This is a little bit disturbing because the literature and everybody expects there to be a correlation, but because few people have studied these tumours in reality, we find you don't see it. So we we set up a model in which we looked at this transition between substrate-independent growth as a community to partial adherence to full adherence, given that is one of the directions of plasticity that you can see. And therefore, it allows us to look for these transition states and look for gene signatures and target presentation that can be important at different points. Very simple flow assay where we're looking at the polysylation of NCAM here. Better to look on this side, the polysylation, where we're looking at the fully adherent form, the partially adherent form, and the non-adherent form. Okay? So here we see clearly that there's a reduction in the amount of polysylation for the early adherent form, but a large expression later on. And that's because this is a competitive environment. And if NCAM polysylation can give an advantage in these communities for proliferation, that will start to dominate the system, right? In other words, it's effectively like a clone overtaking. So proliferation and adhesion compete with each other. You can actually see the polysylation changes here just on an image. Very difficult to image these cells because they're literally like balls of cotton wool, you know, in terms of their 3D structure. But you can see the partially adherent and fully adherent forms all express NCAM, but the level of polysylation has disappeared virtually in the adherent form, which is naturally being generated in these populations. However, there are certain cells here which are highly polysylated, and it's these that you will see actually have a proliferative advantage, and they take over the system. So you've got a dynamically changing system here. And these can change within weeks, by the way, in culture. You think you're culturing the same thing, you disturb how you do that, and suddenly a whole series of new markers have been generated. So if we do gene expression analysis on the glycosylation pathway, we see really no changes in terms of those signatures, apart from a couple of the silyl transferases, which we've yet to explore. But importantly, there is a reduction in one of the key enzymes for polysylation as the cells lose polysylation. So this means that there's been a downregulation of the polysilyl transferase. This is within a background of a whole series of gene changes taking place here within a few weeks, just by the way in which you're culturing the cells. Really quite disturbing, but indicating the extent of gene changes associated with those transitions. And not surprisingly, the early adherent form, that early plastic form, is actually found to have significant changes in cell adhesion molecules. One of the biggest ones that came out of this analysis was actually glypican-3, which is actually highly expressed in some ways diagnostic for some people of squamous cell lung carcinoma, an adherent form that sometimes is found within small cell lung cancer, not as a separate tumor, but as part of that histopathology. So that makes sense. CD44 changes every time you do anything in reality, but CD44, obviously involved in cell-cell interactions, is also significantly changed in that early plastic form. In reality, therapy is targeting this end of the spectrum, whereas resistance arises in this end of the spectrum, when you get highly polysylated, highly migratory, drug-resistant cells. So this is in fact a snapshot of the months that would be undertaken usually for small cell therapy and what it's trying to deal with. If you just look at the different adherence rates, you don't have to look at this detail, just a simple how quickly the cells plate down. As you can see, they virtually don't contact a plastic surface versus partial versus very quickly sit down. Look at the difference in those adherence rates. So this allows us to test whether or not different extracellular matrix proteins are, for example, good targets for some of these adhesion molecules. But in reality, even if you go through all of these on an adhesion type assay, in which you've removed the polysylation on the surface of NCAM just by using a neuraminidase enzyme, so you can remove NCAM, you look at that correlation and you find out that adherence on polysylated or non-polysylated forms of cells that are polysylated not polysilated to start off with, or in fact highly polysilated, don't change. And it's the laminins which are actually the targets for these cells. Not surprisingly, but that correlation means that extracellular matrix adhesion for these cells is not driven by polysylation. Important. It does drive migration though, and we'll see this. In fact, this is a highly dynamic structure. For example, if you grow cells on a hydrophobic surface, and I'm really telling you this so that you have an understanding what the in vitro models can deliver or not deliver. If you put cells onto a hydrophobic surface, they can't, because of the charge structure, they can't adhere, and therefore they form three-dimensional systems anyway. But as soon as you put this down, the cells literally within 24 hours like almost flow out like a glacial flow from these structures. So they're highly plastic in multiple ways and highly reversible. The reversibility is actually important because you could drive a highly polysilated cell into a non-polysilated basin, an attractor, just by changing biosynthesis. The key to this, though, understanding what's happening, is that polysylation does positively correlate with low, medium, and highly polysilated cells for proliferation. It's a proliferation driver. And that's what's driving the plasticity, the dominance of highly polysilated cells within a competitive environment, as you could expect within any tumor. And you can track this by Q-dot analysis, where you're looking for the distribution of Q-dots within a population over time. And it doesn't matter if these cells are on a hydrophobic, a 3D system, or a flat system, it's the same. So in other words, the 3D structure of the community is dominated by proliferation and not adhesion. And that's also seen in vivo if you put these cells into mice. Right, I'm going to pick up the rate of talking now because we've got I'd like to get through a whole series of things. And one of the questions is, what would happen if you artificially mixed populations together and started to drive the direction of a population just by its neighbors? And this is all part of the cell attractor hypothesis. And it's, of course, the ability to detect polysylation in live cells is quite important here. So what we've done is used a GFP model, and I'll just show you what happens as the problem, shall we say, of what happens when... So these are small cells in culture. See this one here? Whoop, charge attraction, captures another group, forms a new microcommunity. I don't know, I think it's this one that might come in. There, reassembles there. That's hopeless for cloning assays. Can you see what's happening? They're aggregating and forming microclusters according to their charge characteristics, and therefore they're forming microcommunities. Actually, nothing to do with clonal expansion, but in terms of assembly within these systems. If you look at a SEM, you can see these the contacts between the cells. What we needed to do was have a new probe for polysylation. And what we've used here is something quite different from usual. It's the same, at least the same specificity, almost certainly higher than any antibody. And that's using an engineered non-catalytic bacteriophage, endosialidase, one which is capable of cleaving surface polysylated molecules. But in this case, it's been inactivated, so the active site binds, but it doesn't cleave. And this is a GFP-tagged bacteriophage endosialidase, endo-EGFP we call it. And because it doesn't degrade it, the GFP, you put it onto the cells, 20 minutes later, or even in live time, you can see the GFP surface tags and which cells are polysylated. So if we go through this confocal Z-scan here, using DRAC7 as a red marker for dead cells, you can see these contacts between EGFP expressing cells very close, but that's about 20 nanometers apart, each one of those cells. So we can track polysylation in live cells. You know, on flow, of course, you can see this beautifully discriminating dead cells here with live cell probes, dead cell probes such as DRAC7. In fact, we use dual excitation of DRAC7 on 488 and 633. And because they give the same signal on both, it means you can put a negative gate around those cells and remove them. It's equivalent of a virtual channel. But I'm showing the plotting of these dead cells here, the highly polysilated and increasingly polysilated forms of the fully adherent versus the suspension form. And when you get to the early adherent form, completely lost this. In fact, if you do high-resolution imaging, though, you will find within that population rare cells that are polysilated. Look what's happening. They're forming dendritic structures as if it's neuroplasticity and are happening within a tumor. So that's the behavioral change that you see upon polysilation. If we just simply mix populations together, this is a little bit worrying, but it is one of the classical tests of attractor hypotheses. So you take, for example, the suspension forms which are expressing polysilated MCAM, and with time, that's what you see. No real change in the dead cell population. So there's the target population. You mix these with cells that are not polysilated, 50-50 mix. Within 6 days, you see the attrition of the polysylation signal. This is going the wrong way. Hope you realize that. You have a highly polysilated cell mixed with a non-polysilated increased population of these, and this appears to drive the polysylation of these cells down. In fact, there's the 50-50 mix, and that's the new population here. In other words, the presence of non-polysilated cells in the tumor starts to drive down the level of polysylation in the over-polysilated cells. Right? So this is because there must be some influence. In fact, to cut a long story short, it's because there's an increase in expression of gastrin-releasing peptide within the suspension form, and this is starting to feed in and give a proliferative advantage to these cells that are potentially adherent, plastic, but in fact they're not growing as fast as they should because they've lost their gastrin-releasing peptide. This is a neuropeptide that stimulates proliferation. So the presence of the highly polysilated cells is giving a proliferative advantage now to its neighbors that aren't polysilated. So this is a complex situation, because if you start to shift the level of the target expression in those populations, you'll start to change the proliferation rates within those tumors. Okay, that's the conclusion. Let's bring this down to a drug discovery point of view where, for example, it's understood that a whole series of tumors overexpress this target, and therefore there's a significant therapeutic opportunity, in particular in neuroblastoma, because this appears to be closely related with the ability to generate bone metastases. As you can tell from the glycosylation pathway, there are certain points at which you could interfere. For example, putting in too much cytidine monophosphate produces a negative signal and reduces the level of polysylation in a target population. Very easy to show. But this is just a tool compound. It's not actually a therapeutic compound. The whole series of novel carbohydrate structures, that's all I can say. Carbohydrate structures have been generated that can suppress polysylation in these populations. And interestingly, they therefore inhibit migration of the cells in the scratch assay, for example. So technically it's possible. These are quite substantial concentrations of these molecules here, but these are sugars. These are just sugars that you're giving to individuals and systems. They're not highly complex xenobiotic molecules. They're just modified sugars, highly tolerated by, obviously, humans. But there's another dimension to this that you should realize. It's not all chemistry and genes, as everything seems to be these days. There's a real biophysical environment within those structures. And that's because you can't get away from the fact that there's a negative charge massively donated to the cell membrane when you polysylate. So there's a cell. This is a biophysics sort of version of a cell. You look at a cell, and as you probably know, it has a nucleus and a cytoplasm. This is how our biophysicists think, by the way, electronic engineers. And the plasma membrane is electronegative due to carboxylate anions and also because of negatively charged sialic acid residues. This is before you start to donate onto the surface massive levels of polysilation. You probably also know, because this is electronegative, it attracts what's known as a stern layer outside cells. All particles do this if they're charged. All cells do this. This is what changes their properties within electromagnetic fields. And this stern layer is present there, and it forms what's known as a double electrical layer. And it's within this double electrical layer, this electronic environment, that most of the receptors that you know about, CD markers included, all these reside in this charged environment. There's a polysilated NCAM molecule. And under those conditions, you allow interaction between these molecules. They interact, homophilic interactions, right, which start to change the way in which those molecules allow other receptors to respond. And the amount of charge that can be added can be enormous by polysilation. There's a fully polysilated molecule, which, as you notice there, doesn't show homophilic interactions because they repel each other. Therefore, in a complex population in which you get highly polysilated and low or negative polysilated cells, you'll have different levels of interactions between cells according to whether or not they allow homophilic contacts or start to reject those at different levels. This is important. So cells find their ideal electrostatic partners irrespective of what they express on the surface of their, in terms of receptors. By the way, we did a study. Can we employ this difference in electronic charge to identify means by which we can separate, isolate, in a probeless fashion, cells that have different levels of polysilation? So we generated microchips, biochips with different electrode arrays in which, for example, here, the electromagnetic field is, electrical field, sorry, is being generated by 4 electrodes here, and you rotate that field so it goes at 90, 0, 270, 180, whichever way you want it to go, a rotating electrical field. And any object within that field will have an imposed motive force on it, depending on its charge. So just by using simple stern layer, double layer analysis, you can determine the extent of that charge just by the way in which these cells interact with a field. So if you rotate this field, the cell will see that field, but the field will change, therefore it turns to it again, then it will change, then it will turn to it again, and you can get a beautiful dance going, basically. There, we're rotating the field, we're changing the frequency now. These cells are changing their rotation speed because the frequency is changing. Eventually you reach a point where there is basically electrical neutrality, and you've measured the charge on the surface of that object, and it's just about there. Okay. So this means you can monitor this very easily, in fact, by image analysis, and you can generate a map like this that allows you to calculate the permittivity of the cell membrane, and by changing the frequency, the extent to which that electrical field goes into the cell, and even the permittivity and thickness, effectively, of the nuclear membrane as well. So all these features can come out. What's the point of that, apart from measuring it? First of all, you can identify highly polysilated cells, but because there's, in this case, a difference in cytomembrane permittivity, a little like the ability of you to pick up a phone signal from inside here because of the nature of the walls, the extent to which the radio mast is trying to penetrate into there with different frequencies. So this means you can invent ways of separating cells based on their polysilation. I'll give you an example of that. There. See, cells separating on just 2 electrodes according to their differential charge. And by the way, they're under-polysilated and highly polysilated cells. So that's really to tell you, and this is the people involved in this, not least Bangor University, that did a lot of the electronic engineering and collaborations with endo-GFP people, and also the drug discovery people of Bradford University, in particular Robert Faulkner. Let's get down to hypoxia, because I'm getting a little bit breathless anyway. So really, hypoxia is linked with pH control in the cell, and also the extent of vascular access. I want to quickly touch on the relevance, the sensing of it, how you can target it by design, by prodrug technology, in particular targeting DNA topoisomerase within the cell as a cell cycle target, and some of the in vitro and in vivo models. So I'll quickly go through this because you should understand this, because it's one of the basic things in biology, and I'm sure you do. The pH profile of tumours can be as low as 6 to 6.5. And under these conditions of acidification, a whole series of changes take place in the cell, not least, for example, the sequestration of certain molecules like VHL, preventing it from targeting HIF, the hypoxia-induced factor, for degradation. So you have this situation where an acid environment starts to contribute to a metabolic shift, favouring the maintenance of hypoxia-responsive elements. Right? So in other words, this effectively is a hypoxia feedback loop. So as soon as you start to get acidification, you start to stabilise HIF, then you start to promote the ability of those cells to live differently and more distant from vascular supply. It becomes an iterative process. This is why tumours can outstrip their vascular supply. One of the key factors for acidification, of course, is carbonic anhydrase 9, often used as a marker for progression of tumours during therapy. You can pick this up in the blood. pH regulation and ion transport in many organs is controlled by carbonic anhydrase 9. Not surprisingly, it's a membrane-associated glycoprotein. And by intensifying the extracellular acidosis, this facilitates, through CA9, the activation of proteases that start to degrade the extracellular matrix. So in other words, you get acidosis, allowing cells to become more robust within a hypoxic environment, and they start to degrade the extracellular matrix and stimulate migration. Because they've had enough of it there, they need to move on somewhere else. It's a self-perpetuating process. In fact, you can design probes to actually see inside cell. Here's a near-infrared marker that's a derivative of CA9, allowing you to identify CA9 expression, acidosis within tumours, for example, here in a mouse model. So, as you know, we need markers for hypoxia to understand what that environment, where it is, and the extent of hypoxia. It's not surprising that, however, you need to vary the amount of oxygen in a system to test this. Now, this is, you should be aware of this. 21% oxygen is toxic to many cells. So the incubators that you are running are mostly putting the cells under a large degree of stress at 21% oxygen. So for example, if you just take, for example, a P52 a BCL2 overexpressing versus a BCL2 overexpressing P53 negative, that's a P53 positive, you just change the oxygen supply to 3% in the incubator, and all the background of dead cells disappears, and they increase their growth rate by about 1.5 to 2-fold. So most of the time, working at 21% is a toxic environment. And if you look at the level of oxygen from 21% down to 18%, down to 14%, you get through to a tissue, about 10%, you get around to the surface of a tumour, 5%, inside the tumour, 3% and below. So remember, to study hypoxia, the controls are sometimes in a toxic environment. If you think about the ability of oxygen to even penetrate into microcommunities, you can model this, and the basic outcome of this is that you can identify within three-dimensional tight spheroids, for example, and an anoxic, hypoxic, and oxic layer. You're obviously aware of this. And this is why anoxic cells die, and you see this clustering of anoxic cells, necrotic cells within the center, a hypoxic surface, and this level here is only about 200 microns. Beyond 200 microns, oxygen is really significantly inhibited in terms of its passage into those communities. It's obvious that within a complex structure, as we see in tumours from the primary site, where there could be low levels of oxygen, but there's also evidence that microniches to which tumour cells migrate are already hypoxic, certainly within the bone marrow. So it's a homing influence as well. Why do we worry about hypoxia? Well, there are lots of origins, but the impact is, first of all, it occurs in poor vascularized regions, and therefore drug supply is bad. Metabolism can be changed of the drug under hypoxic conditions. Radiation therapy depends on the radiolysis of water in the presence of oxygen to work. Therefore, hypoxia produces high levels of chemoresistance and radiation resistance. But therefore, there's an opportunity to overcome this if we can use the hypoxic environment as a targeting influence. There's a whole series of probes that you can use, not least hypoxy probe, nitroimidazole, that becomes triggered to bind when you have low levels of oxygen. You fix the cells, immunostain, and it's a good indicator of the cells that experience hypoxia. At the clinical level, positron emission tomography is the word at the moment to try to identify hypoxia directly within tumours. But many of these, for example, here, and to some extent here, and certainly there, are based on nitroimidazoles, molecules that can be bioreduced to a different active form and therefore have residence, almost certainly because of the acid of environment in a tumour, and allow you to see hypoxic regions by imaging. And it's an example of one of those. But there's a whole series of requirements for designing a drug that actually goes in and identifies a hypoxic region and becomes activated. I won't go into that. Let me tell you that there are different types of drugs that have been tried before, and this is a long history of failure. When you have a target that you know is there, is available, and can be exploited, and will overcome drug resistance and destroy many of the important tumour cells from which a tumour will relapse, you still see failure. And the failure is human. It's because of the need to run a trial without selecting the patients that show hypoxic tumours. In other words, no stratification. And it's been a history of failure up to now because of either the drug itself isn't quite right because of toxicity, or because you haven't selected the patients that show the hypoxic tumours. Isn't that just obvious? But we're talking about 25 years now. One of the problems is, of course, that the pharmaceutical industry quite rightly wants to give the drug to everybody, irrespective if you have a hypoxic tumour. Therefore, when you analyse, such as in the case of TH302, just recently failed in pancreatic cancer in, December 2015, that's because they didn't really select the patients. And they understood that. But the attempt was to try to widen the use of this drug, obviously because of pharmaceutical imperatives. So we're looking at a new class of drugs called aliphatic N oxides with the concept of taking a very potent anti-cancer drug, inactivating it to a prodrug. All drugs should be prodrugs. In other words, essentially non-toxic, but only work at the site of action, rather than being systemically active. And then we take that and we modify the structure of that molecule so it's electronically neutral, so it passes through the whole of the body. It even passes the blood-brain barrier. Very important, for example, for brain tumours. And we've got two candidates here, OCT1002, OCT1004. And basically these are, in the potent form, intercalating molecules that bind the DNA with high affinity. And I don't know if you know this, drug design is based on using a pencil. Really is. It's very sort of, you'll see the pencil appear to indicate, first of all, that there are active areas of this drug here and here. There it goes, which tells you that these are the hypoxia sensing N oxide molecules, which are bioreduced upon hypoxia, allowing this part of the molecule here to intercalate into DNA. So this is electronically neutral, non-intercalating, then it's activated within the cell to a potent form. This is a 2-electron reduction process for each one of those side chains, and therefore it's a 4-electron reduction process. But importantly, compared with every other drug ever put forward to target hypoxia, it's irreversible. The point is, if you start to reoxygenate, what happens to those molecules? And what happens for nitroimidazoles? They generate superoxide radicals, etc., which start to damage non-target tissues. And that's why you see toxicity. Think back to the very first talk now in this course, right? Talking about superoxide damage. Many of the drugs that have been used before are actually toxic because they're generating these radicals. This is quite different. This is irreversible. So in other words, it's like your credit card. Once you see it, you know what you've bought and you can't get away from it. It's a molecular ratchet, as you call it. So I'm going to quickly go through now the fact that there are advantages for blood-brain penetration. It's controlled by cytochrome P450, and these molecules are turned over by cytochrome P450. Of course, if they're not there, the right cytochromes, you don't get the activity. But virtually all tumours have all of these cytochromes in place for activation of this prodrug to its active form. But you have to be aware, if you start selecting cell lines to look at this, cell lines are very selective in terms of the cytochrome P450s or heme-based molecules that they express. So you can get, for example, cells that express the right type of cytochrome P450 and some that don't. So choosing the right test system is really important. So when we do this, we see generation of the active form of the drug commensurate with the amount of hypoxia in a test system, virtually no conversion here. And this is what, approaching 100-fold when you have just 100 nanomolar of a non-toxic drug being converted to an active form. In fact, the stoichiometry of this is so good that you can, because the molecule itself is intrinsically fluorescent, by the way, in the near infrared, you can just measure the amount of accumulation of fluorescence and you can calibrate your incubator based on that. But you see the difference between no conversion under normoxia, massive conversion under the level of hypoxia that's relevant. The drugs themselves, once activated, accumulate within cells, here you can see this directly, and move to the nucleus and target topoisomerase II, where they induce DNA damage, as shown here by looking at gamma H2AX. Simple proof of principle. They are anti-proliferative as a result because of the induction of that damage specifically in hypoxic cells, and you can look at that using a whole series of techniques, for example, not least the ability to see changes in apoptotic levels as cells respond accordingly as the drug activates. There are a whole series of issues now, because in these environments, sometimes cells don't die. What they do is exit the cell cycle. They go into long-term quiescence. So therefore, looking at, for example, cell death, apoptosis induction, for these long persistent drugs is inappropriate. What you're looking for is a long-term inhibition of the cell cycle. There's a whole series of ways that you can do this by using tracking dyes, for example, that allow you to, this is cytochrome, cytrac orange, that allows you to see this sweep up here of cells into an enlarged state on the presence of hypoxia and the drug compared, for example, with the control, where you're looking really at the size of the cell, because the cell is sat there, out of cycle, same amount of DNA, so you don't normally see that on cell cycle arrest so clearly, but the cells are getting larger and larger. Something interesting is happening there. They're still producing cytochrome P450s. They're large drug factories. So the concept is to put the cells into these states. The drug is being produced into an active form, it's overloading into these cells, and guess what? It has a significant bystander effect. So you're getting two hits. You make it turning the cell itself into a factory for your active drug. Okay. There's what you'd expect under hypoxia, and the prodrug, you get the cell cycle arrest in G2. I'm going to skip through this very, very quickly. One of the reasons is I like it, and I want to tell you about it, and it might elicit questions later, or in fact approaches to me separately by email, right, if you want to know about this. It's a very, very useful probe. It's using a probe which allows us to track the cell cycle in real time, consistently over long periods, to the point where you can predict what is about to happen next by looking at a cyclin signature. And this has been used because there are unusual activities, actions associated, mechanisms of action associated with drugs that inhibit topoisomerase 2, not least the ability to inhibit a checkpoint late in the cell cycle here called a decatenation checkpoint. You know, there's the checkpoint that is, when it's disrupted, does not allow molecules, chromatin molecules to segregate, and therefore they go into a permanent arrest, or they go through mitosis and fail in mitosis and they kill the cell. Right. So if you inhibit this, for example, there's different ways of doing that in inhibition. Here's with a catalytic inhibitor called ICRF193. It blocks the topoisomerase molecules' ability to decatenate DNA. Importantly, it's P53 driven, this process. I won't go into that in any detail, but it's P53 driven because it's linked in with the stress response of cells. Let me just get through to this. This is the important thing. How do we track transition of cells through checkpoints and what their eventual outcome will be? Because there's a hidden story here. Not all cells go into polyploidy, as you realize, through mitosis. So cells are capable of sidestepping all the mitotic checkpoints by going into an endocycle, appearing in G1 polyploid, and if you look at the cells, they're doing nothing. They're just blinking at you. They appear to be arrested in G2, but they've actually transitioned into another state from which they can generate aneuploidy without ever going through mitosis. And because you need trackers for this process, you can use cyclins, in particular cyclin B, the signature that arises and is destroyed in mitosis. And by using a stealth reporting, which we have GFP associated with the cyclin B1 promoter, with its destruction box, but critically it is not active. So this reporter does everything that cyclin B does, and it appears, but it stands behind cyclin B like a shadow. It doesn't participate in any of the reactions, but it's produced, translocated, and destroyed with the same kinetics. Now this is important because normally what you do is tag active molecules, and they interfere with the process because you're overexpressing. So this is a stealth reporter. This is quite revealing. So there we have the stealth reporter in osteosarcoma cells. A G1 cell there, I can just see this, goes into S phase, more cyclin B. It translocates it to the nucleus, then destroys it. 2 G1 cells that are dark. Here's another one. I can guess that's about to do, right? That was very quick, a bit too quick for me, really. So this signature allows you to even guess if a cell is about to divide or not within a few hours. A G2 cell increases, translocates, destroys. Right? So you can get a full cell cycle signature of individual cells now in a large mass, looking for synchrony, asynchrony, specific induction of arrest. You can look also, because you can track live cell die here, DRAC5, with mapping this with cyclin B. You see subpopulations here, and this is a critical one. You're used to seeing cells going through G1, S, G2, then back to the start again. Cells can go G1, S, G2, cyclin B reduced, but still in G2, then go into polyploidy without ever going through mitosis. In other words, these cells are endoreduplicating, but you can't see it normally. This allows you to track these populations with time. By the way, these cells are viable. They resolve into different aneuploid clones just by bypassing this checkpoint. So importantly, because it's P53 driven, this process, if you have, for example, dysfunctional P53, what happens, cells ignore this commitment to bypass, and they go through a furrow regression. You get binucleated cells, or if they escape, they'll go back to the start. However, in functional P53, what you get is a commitment to this bypass sometimes 75% of the cells doing this, but it's unknown in the analysis usually. And you get this route to chromosomal instability that is occult. Where you put cells under hypoxia, therefore, you can see different cell cycle arrest signatures according to when you challenge with one of the drugs that we're looking at here, whether or not you go into endoreduplication, or you, for example, here, going beyond G2 levels, or basically you start to arrest. So, last 5 minutes, how do we target the hypoxic environment? Because at the moment we're in the position of trying to put a drug, one of our lead drugs, into a phase 2 trial supported by hypoxia imaging. I'll just give you a little bit of insight into some of the work that was done in the preclinical phase. You have to choose which tumour you're interested in, obviously. And probably the best ones at the moment are prostate cancer and pancreatic cancer because of the high levels of clear hypoxia that's shown in those tumours, with the concept, of course, of targeting these cells, these hypoxic cells, with a prodrug, which is very rapidly cleared in its non-activated form, leaving the potent agent in those regions, and therefore more fully completing the therapeutic intent. Clearly, the toxicity of the prodrug itself is important, so screening assays were done to identify the limits at which you could use this. This starts to inform the in vivo dose models. And this is one of the first things that we did. We asked the question whether or not therapy, currently standard of care, could induce hypoxia, and it does. Otherwise, I wouldn't be showing you. And a good example of that is the use of an anti-androgen in the treatment of prostate cancer, in which, for example, typically a person is put on, all males for some reason, I don't understand that, anti-androgen, basic misunderstanding of the biology here, anti-androgen therapy in which those people are put on that treatment before, for example, radiation therapy. Now, if that's producing hypoxia in potentially migratory and invasive cells, that's the problem, clearly, for later therapy. And in fact, for any therapy that targets proliferating cells, such as gemcitabine. So from that point of view, what happens when you put cells under androgen deprivation? Well, first of all, BCA there, biclutamide, also known as Casodex, reduces growth. Everybody's happy with that. It's an anti-proliferative. If you combine it with a hypoxia-activated drug, you see this synergy here very nicely. Fine. These are all colony assays. That's fine. Exactly just what you'd expect. There's a cell death marker associated with that combination. However, let's explore what's happening in a tumour situation by using a probe that can identify hypoxic regions. One of the biggest challenges, as you know, almost certainly over the years, is how do you measure hypoxia, just the state of a tumour. This has been a real problem. One of the original techniques was using the Eppendorf electrode, which is equivalent of, you know, it's just like putting a garden fork, basically, into a tumour. It really is difficult to get meaningful results, although people have produced some excellent results in good hands. Here's an example that we use, an oxylight sensor, which is a fine probe that goes inside the tumour and senses the hypoxic environment in a way that's commensurate with the level of partial pressure of O2. When you do that in a biclutamide-treated tumour, you see something amazing. It immediately starts, as the tumour grows, to generate a low level of oxygen, and then it recovers. Quite expected. You can expect this. What happens? It starts to disrupt vascular supply. The cytokines are released, and you get a rebound. And from there to there is what you see clinically. What is actually happening in the tumour is that. So there's a window of opportunity here for targeting hypoxia. And of course, when you do that, you give a relatively small, non-toxic dose of a hypoxia-activated drug at the right time during that biclutamide model therapy. You see, you knock out the tumour growth completely. In other words, therapies induce windows of opportunity because they dynamically change the proliferation rate and vascular supply of a tumour. They change that, and it gives you an opportunity for therapeutic targeting. Of course, you have to see what's happening. So here's a window analysis. Quite heroic experiments, I always think, certainly for the mice. When you generate a window in the side of the animal, so you can use microscopy to look at the vascular supply of tumours as they're under treatment. For example, with the hypoxia-activated drug, obviously some disruption there because you have hypoxia targeting stromal elements as well as tumour here. Bicalutamide disruption, then it starts to come back. Combination changes this completely compared with that. I won't go into the analysis of this. This is looking at the branching structures and size and length of the blood vessels, but it's very obvious that combination with a hypoxia-activated drug during the course of bicalutamide therapy starts to disrupt the vascular supply and also knocks out the ability of the genes that are involved in revascularization to be activated. So, important principle here, is the drug there at those locations, or is it a distant effect? Because there's a natural fluorescence associated with the activated drug, you can find out that the non-vascularized regions have high levels of drug in those distant from the blood supply. In other words, the drug is activated, and this is, by the way, after a single dose, at 1.5 days, at 14 days later the drug is still there. It's one of the most persistent and active anti-cancer agents that we know of. What's more importantly, during this period of hypoxia, the presence of a hypoxia-activated prodrug knocks out metastasis. So there is now the horror that anti-androgen therapy for prostate cancer induces migration, invasion, and metastasis. In other words, the treatment itself is promoting that. And we're suggesting that a hypoxia-activated drug targeting the microenvironment allows that to be knocked out. And the same works for pancreatic cancer as well. So, I finished. Thank you very much for your attention. And there's a future here for combination with the whole series of agents for using drugs that target the microenvironment, microcommunity interactions in a whole series of tumours. And I think that represents the hierarchy of our priorities at the moment. A whole series of people involved in both the drug and probe design sides, certainly from University of Ulster and Cardiff University. I'll pick out Isaac Treasurer because I have to, otherwise she'll kill me. Rachel Errington, for example, who did a lot of the imaging work. We have clinical teams in Toronto that are picking up the potential here for trials. I want to thank you for listening. We got through a lot of slides there. It was 190, by the way. Okay, so give yourself a clap. That's enough. There's nothing worse than a premature self-congratulation, as they say. So many thanks to our hosts, Andrea and Paul, and also to Thermo Fisher. A little wave. Go to the Bioprobes catalogue. It's not the Bible. I refuse to accept that. It's the New Testament. Right? Because there's a new field out there of smart probes. Go to there. And if you don't mind, I'll just leave you with one insight into the frustration we've had over the years in trying to design probes and also drugs that target the microenvironment. Right? And that's because there are just too many problems. And one of the essential problems, you design a drug, by the way, our lead drugs are it, we think. There's no more development required. If this doesn't work, I retire. You design a beautiful agent, and you've got the molecular target, and you're chasing that. And this is an insight into what it's like. There's the beautiful agent, perfectly designed. There's the target. It's in its environment, specific. Then biology comes along and starts to disrupt the possibility of that happening. And before you know it, the target has completely disappeared. And it's understanding those dynamics that's going to be your problem. Thank you.
Thank you, Paul. Wonderful talk and wonderful final. Any question? Paul. Paul to Paul.
Thanks, Paul. That was really a delightful presentation, and I learned a lot. When you started talking about the difficulty of using in vitro systems to represent biology, it occurred to me that there are ways of solving this problem. And I'll sort of ask you a question and then pose a potential solution. What models have been developed, what synthetic models have been developed to understand the deficiencies of each in vitro system? And I'll give you one example of where we no longer do in vitro studies. In the world of nuclear bombs, we don't blow up bombs anymore to understand what the bomb design is going to do. It's all done with supercomputers. And the question is, why is there no major drive to apply that to our understanding of different models? Because at the end of the day, we don't have a lot of choice in that we have to do in vitro studies, but they're so deficient. How can we accommodate that? It's a sort of a struggle. You have to use the microphone.
Can you switch on the microphone?
Hello?
Yes.
A long-term relationships in the nicest scientific form, and with mathematicians, engineers, electrical engineers, etc. Right? Good example of that is actually being able to mathematically model, for example, complex cell cycle data where you're looking at branching structures, which is a real challenge mathematically to see how you have a model that allows you to look for branching structures. So converting, as we all do, biology to numbers, but putting those into predictive models is certainly one of the solutions, first of all. You have to realize what I've been talking about is not the sweat, it's more of the hypothesis-driven side of drug development. The sweat is actually having a million compounds and trying to work out which are the right ones, as you know, right? So I've ignored completely all of that process and just jumped into what we think is the idealized molecule to take that forward. That's fine. So the problem is, what you lose in those systems that you're talking about that don't fully fulfill the needs for presenting the target in its appropriate environment, and what's the attrition rate, and it's absolutely enormous. You know, if you calculate the amount of sweat and effort, and that's why it costs, what, 2 million for a phase 1, 20 million for a phase 2, 200 million for a phase 3, 750 million to turn that medicine into a global market, 1.2 billion required to return that drug to some sort of profit for a company. The attrition rate is enormous. So the question you're really asking is, how do we indicate in that process, not more and more relevant systems, in AstraZeneca realizes this, but how do you indicate early failure? Early failure. So you challenge the system with what you think is a critical activity for limiting the drug activity, virtually always toxicity, and therefore it's irrelevant to have liver, kidney, brain models that highlight toxicity on non-target tissues. And that's probably the best way forward, not looking at the tumour itself, because in the end, most drugs fail because of a failure of inappropriate targeting, toxicity, not able to reach the maximum tolerated dose, non-compliance, side effects, the whole series of things. Quality of life as well now, certainly for funding within the UK of the therapy. So early failure is important. So in fact, people should really be working most of the time on normal tissue models for toxicity, right? Now, that's awful to think that way, but it reflects really what Mario was telling us. In other words, models for looking at, you are sitting there, Andreas, he's gone. So you should be looking at normal biology and how that can preclude you using that targeted therapy. But I don't really believe in my heart, but it's fun to do the mathematical modelling because they always simulate rather than predict. But you remember, I haven't been talking about all the sweat beforehand of trying to choose the right drug in the right target. I've gone right to the other end, just showing the cytometry. So, but failure, I think more focus on normal tissue systems is the way forward. And that's what all the grant calls are now for example, in the UK, for pharma-backed projects or research council, understanding normal biology in context, not the tumour biology.